I'm going to talk about protein synthesis. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. This copying process is known as transcription. It occurs in the nucleus and is the first step in protein synthesis. The second step is called translation. A portion of the DNA is unzipped, so the mRNA can be made from the DNA like a template. Nucleotides from the RNA match with the ones from the strand of DNA and make mRNA, also known as messenger RNA. RNA polymers unzips the DNA and puts the nucleotides into place. C and G nucleotides match up, and A and T nucleotides match up. But every time RNA places a complement on the A on the DNA, it places a U instead of a T on the RNA. The mRNA doesn't copy a whole strand of DNA. It only copies the portion of the DNA for the protein it wants at the time. The small section of the coding DNA has a start and a stop signal. After the DNA forms, it moves out of the nucleus through the nuclear pore and goes to the cytoplasm. There are 3 types of RNA. The purpose of mRNA is to take the protein code out of the nucleus without pulling the DNA out of the nucleus. mRNA will need to find a ribosome in the cytoplasm. mRNA is read 3 bases at a time. The 3 base pairs are called codons. The ribosomes are made of protein and RNA. Ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. tRNA is transfer RNA. Its purpose is to transfer the amino acid. It has an anticodon on one side that will match with the codon on the mRNA. Together, these make translation happen. Translation occurs in the cytoplasm where there are ribosomes. To get the translation started, mRNA attaches a ribosome and a codon that must be read. Usually, this starts in AUG. The next tRNA molecule moves in and matches with the mRNA codon while the amino acids form a peptide bond. The previous tRNA detaches and the mRNA shifts and moves for the next tRNA molecule to come in. The protein grows until a stop codon is reached. The protein is formed and ready to finish folding to become functional. Once the stop codon is reached, the protein is formed.